Welcome everyone to the ELIXIR GA4GH workshop, ECCB 2020. My name is Gary Saunders. I'm the Human Data Coordinator at ELIXIR, and I'll try and chair this session along with my co-panelists who are all presenting later and have joined through the Zoom window. So some housekeeping notes before we start. This workshop will run in webinar mode, so that means that only the panelists will be able to speak in the video window. But in order to be as interactive as possible, we do encourage as much input from attendees as possible. So please do use the chat window in the open air platform to ask questions and initiate discussion. So it'll be my job to monitor this chat as much as possible throughout the event with the help of the co-chairs. But if I do miss anything, or you would like to discuss anything further, my email address is there, gary.saunders@elixireurope.org. So as an introduction to today's session, I'd like to introduce you to ELIXIR and then the challenges we face in the human data field, what we're doing to address these challenges, and then some conclusions that will be a gateway into the rest of the session. So, who are we and how are we structured? Well, there are hundreds of ELIXIR institutes across Europe that have signed up to join the research infrastructure. And these institutes come from 22 member states of the European Union, plus EMBL-EBI. And there is one hub for ELIXIR that's based in the south of England and Cambridge in the UK. And ELIXIR works in a hub and spoke model. So where we have one hub, we have connections to all of our 23 nodes. So the objective is to take the institutes that are part of the ELIXIR infrastructure and find common goals of interest so that we can provide a coordinated suite of services for researchers and clinicians across Europe and indeed wider. This coordinated suite of services and this coordination of the institutes, this is ELIXIR. That is the research infrastructure that I represent here. So the way that we work internally is that we have 5 platforms represented by the orange circle here. So we have tools, compute, training, interoperability, and data. And around the outer side, we have a number of use case driven communities. And if we look at these gray lines in the middle, this represents the interaction between the communities and the platforms. So we have a number of activities or projects that link the use cases from the communities to the services that are offered by the ELIXIR platforms. So in today's workshop, we're focusing on ELIXIR and GA4GH. So we're really looking at the human data challenges that are posed to European infrastructure for managing sensitive human data. And where we work right now is in the space between research data meeting the healthcare space. And this is represented by the numbers here. So this is the percentage of human genomes and exomes that are funded solely by the healthcare systems. So back in 2012, it was as little as 1%. In 2017, this moved to around about 20%. And the prediction is that in 2022, as much as 80% of the human genomes and exomes that are generated are funded solely by the healthcare systems. Now, healthcare data is a national competence, so often these data are held much more securely than data that's generated in the research field. So often data that's owned by the healthcare systems can't leave national jurisdictions. So in order to access these data transnationally, we have to try and overcome these challenges. You can see the manifestation of these numbers in this slide here. So this is genomics-based national initiative projects across the ELIXIR members. So nearly every ELIXIR member has one or more national initiative projects generating genomics data within the healthcare system. So these national initiatives will be the data generators and the data owners of this genomics data, which for use cases such as rare diseases, we have to try and access and analyze across national borders. So this schematic is something that maybe we'll come back to time and time again throughout today's workshop. And this is the vision. It's the federation of data essentially across the globe, where each of these blue dots represents a sensitive data archive. And if you look, these blue dots are connected together. So they're connected together with a suite of interoperable standards. So we need the same standards to be adopted by as many of these blue dots as possible so that we can access and analyze these data transnationally. So what are we doing from ELIXIR, from GA4GH to try and address these challenges and build this federated data network? Well, there's willingness to try and, first of all, there's willingness to try and address these challenges. So a couple of years ago now, a number of countries within the European Union signed a declaration to access at least 1 million human genomes by 2022. And on the side of this slide, you can see the map that there's a huge overlap between the countries that signed this declaration and the ELIXIR member states, the nodes of ELIXIR. So there's a mandate there for ELIXIR to take a driving role in building this federated data network. And one of the first things we did from ELIXIR was publish this paper in Nature Reviews Genetics. And this has contribution from many of the key stakeholders across the ELIXIR nodes and wider about the core infrastructure elements that we need to access 1 million human genomes by 2022. And these are summarized in this table here. So I don't mean to go through this in any great detail. The paper is online, it's open access. You can, please do encourage you to read that paper. But essentially a number of these minimal infrastructure components are associated with the Global Alliance for Genomics and Health, the GA4GH, which is an international standard setting agency. So ELIXIR has aligned closely with the GA4GH, not only to implement the suite of standards that are generated, but also to help develop the standards and use the ELIXIR GA4GH strategic partnership as a gateway to implement the GA4GH suite of standards where there is a lot of European contribution already, but really use their strategic partnership as a gateway to try and drive the implementation of this suite of standards within Europe. So this is a bit more detailed than the blue dots that I showed earlier. This is how we view the federation of human genomic data. So there'll be a number of national-based sensitive data archives that will allow the data to be stored locally. And then we'll have a suite of interoperable standards that allow us to access these data transnationally. And in here, I've got things like ELIXIR AAI. I've got logos for the European Genome Phenome Archive, the EGA. These are things that I won't go into in depth in this introduction, but these are tools and services and components that we'll hear much more about throughout this workshop. So ultimately, the system relies on 3 elements: discoverability of data, archival of controlled access data, and then cloud computing environments to analyze these data transnationally. And if we take these three key components, then at the bottom, I put a suite of the GA4GH standards against each of these elements. So those that don't work within GA4GH right now or are aware of GA4GH, this might be quite daunting, and I don't mean to go into any of these standards in any great detail, but in the next couple of hours in this workshop, the presentations that we have will touch on many, if not all, of these standards that are described in this one slide. So in summary for an introduction, ELIXIR is developing a transnational, interoperable, sustainable infrastructure to manage sensitive human data. And the policies, roadmaps, and technical standards that are needed here, a lot of that is done in collaboration and in alignment with GA4GH. And the objective right now for this presentation and for this workshop today is to show how we are aligning with the key stakeholders across Europe and indeed wider to try and unleash the possibilities for genomics and health across Europe and wider indeed. So this is the schedule for today's session. I've given an introduction. We'll then move on to Dylan Spalding, who will give us the view of federating services from the EBI, the Emble EBI based in Cambridge in the UK. We'll then have a second presentation from Emble EBI with a slightly different focus, coordination of metadata standards from Melanie. And then I'm really pleased to see that we have Mamana on the schedule representing H3 Africa, who'll give us the international view. And then we'll have a deep dive into 4 key use cases that ELIXIR focuses on: rare diseases, cancer data, complex disease, and infectious disease. And then as the last technical presentation of the session, Shubham from ELIXIR Finland will give us a presentation from the ELIXIR GA4GH Cloud and AAI driver project, demonstrating how we're providing cloud resources to access and analyze data across borders. So with that, I think we can pretty much get started. Hopefully you can all still hear me, and if there are any questions that have come up in the discussion forum, I can take them now. It doesn't seem like there have been. Then I pass it on to Dylan, who can take us through the first presentation. Dylan, you can start to share your screen, please.
I hope you can see my screen. So I'm Dylan Spalding, and I work at the EGA, the European Genome Phenome Archive, at NTB-EW02 in the UK. And I'm going to talk about how we're federating the currently centralized services that we have. So most humans, as Gary has said, most human genomes were previously generated by research projects, but now the balance is changing so that more genomes will be generated via national healthcare initiatives. This increases the number of genomes available with the accompanying capacity issues, but it also has ethical, legal, and societal issues regarding the transfer of the data between these various jurisdictions. The danger here is that the data will become and remain siloed, which will reduce the data's usefulness for research. The European Genome Phenome Archive is becoming federated to try and help deal with these issues, and I'll use it as a use case for describing federated data and the standards being implemented and used at NTB-EW02. So the EGA is a joint service from the European Bioinformatics Institute in the UK and the Centre for Genomic Regulation in Spain. And it aims to be a secure and permanent archive for all types of genetic, omics, and phenotypic data for humans. All the data is consented for some form of research use. And to gain access to the data, you must apply for it, and then the data is distributed to you in an encrypted format. We are a GA4GH, or Global Alliance for Genomics and Health driver project, and we're also an ELIXIR core data resource. Just to give you some statistics on the scale of the EGA, currently there's around 11 petabytes of data available within the EGA, and we accept submissions on a global scale. We also distribute data globally. So far in 2020, we've distributed over 4 petabytes of data, and these data are held in almost 2,500 studies and over 5,000 data sets. So the EGA supports a large range of interfaces to support its federated vision. These interfaces and APIs support the website, the submissions of controlled access data and the associated metadata, the permissions management, the data distribution and analysis, for example in a cloud, project-specific portals, and the federated EGA itself, which I'll concentrate on the next slide. So federated EGA aims to be a harmonized platform for human data discovery, access, and distribution. This will help deal with the scaling issues I mentioned earlier, where the number of genomes being generated is expanded, and also the ethical, legal, and societal issues related to that data, such as GDPR. In the federated EGA, there's a centralized metadata model, so that only the controlled access data is held at a federated node, and that federated node distributes the raw controlled access data. To support this, there are 5 main types of API: the permissions API, a data transfer API, the submission API, AAI, and metadata. It turns out that most of these APIs are covered to some extent by standards being developed by the GA4GH, and hence the EGA tries to align the development of its APIs with the standards created by GA4GH. One of these standards is the Authentication and Authorization Infrastructure Standard, or AAI, which has been defined by GA4GH in collaboration with ELIXIR and is implemented by ELIXIR as the ELIXIR AAI. This AAI is interoperable between ELIXIR nodes, supporting single sign-on. As it is GA4GH compatible, it supports linked identities, which I'll explain in a minute, and a user may link their EGA identity to an ELIXIR identity, allowing the user to access EGA data using their ELIXIR identity. This single sign-on and linked identity feature of the ELIXIR AI underpins federation, allowing different nodes to administer their own permissions, but via a single user identity. At EGA, we've developed a service which allows the users to link their ELIXIR and EGA identities, and the linking can be visualized using the ELIXIR GA4GH ECHO service. Here's just an example of a linked identity and a list of controlled access grants. One can see that the EGA identity is linked to a specific ELIXIR identity in the bottom paragraph here, and that the EGA identity has access to at least one dataset within EGA, which is in the top portion. And this is an example of a GA4GH passport specification, which is a standard developed by GA4GH. Another standard implemented at EGA is the data use ontology, which classifies the allowable data use for a dataset across resources, harmonizing the terminology used. One advantage is that it enables its discovery of data based on the research use, i.e., for example here, data that cannot be used for research into brainstem glioma can be excluded from the results set. This can then be used to help automate data access as the ontology is machine readable, and I'll describe how that's happening later on. Another standard that we've been implementing is phenopackets. In a federated network, there's a requirement to share phenotypic data, and ideally these data should have a consistent representation. So we use the phenopacket standard developed by GA4GH clinical and phenotypic data workstream, and it has defined a PXF file format for exchanging these data. It can support detailing the phenotypes, disease, and individual attributes, and it is ontology based. It's also interoperable with FHIR. EGA accepts phenopackets, and this will ultimately help improve the harmonization of the phenotypic data based in EGA in a federated EGA network. Here is an example of an anonymized phenopacket which was submitted to us via SolveRD, which is a rare disease use case. And here you can see the details of the subject, which is female, has multiple phenotypes associated with it. When we take these two together and link them to BioSamples, you can actually start to really see the power of extending the findability of controlled access data sets. So EBI resources tend to use BioSamples to try and link the data between the different resources, and EGA is no exception to this. So aggregate level data that isn't personally identifiable, but the raw data which may be in EGA and could be in an open access resource such as EVA, can be linked via BioSamples to EGA. Now the phenopackets allow a consistent way of representing the phenotypic data, whether it's in either the open access resource such as BioSamples or in EGA. And additionally, BioSamples also now supports Duo. So users can actually log in, access BioSamples and search for data sets which are in EGA where the samples have certain phenotypic features and that have a certain data use. Another way of improving data discovery is via the beacon. The EGA currently has a beacon which gives a binary answer to a query on whether a certain allele exists within a resource. But we're working on the next version of the beacon which will extend the queries to SNPs, structural variation, and regions. It will also add filters. So again, you can filter by sample attributes, data use, and individual attributes. Also, the responses can be tailored depending on the access level of the user. So for example, you can increase the amount of information in the response if the user is at a registered or controlled access level, i.e. have logged in via the ELIXIR AI, so you know the user who is making the request. I've mentioned GEO, but the real power of GEO and permission is when you consider the scale of the number of DACs or data controllers that the EGA supports. We have over 4,000 data sets, each of which has one or more DAC controlling it. And each of these DACs have diverse range of data use conditions they apply to the data sets. And we have over 17,000 users accessing these data. So therefore, we can see that to speed up the access and facilitate the access to these data, some form of machine readable way of representing the data use and the identity and ultimately the permissions of the user accessing the data is crucial. So here we combine the AAI, the GEO and the passports, and these hold the information on the identity of the user, the institution of the user, the bona fide status of the user, and the proposed data use that that user wishes to make use of the data. And this can help facilitate the data access decision making. For example, there is the data use oversight system, which has been developed by the Broad Institute, and this uses GEO then to try and predict the data access decision made by a DAC on a particular data set, and then the DAC reviews that decision and makes their decision. There is also the Resource Entitlements Management System, REMS, and this is a federated system which allows a user and a DAC to log into a particular service, and that can grant and revoke access to data sets in a federated network. So you can, REMS could apply permissions to EGA data sets and allow DACs of EGA data sets to grant or deny those permissions. Finally, there's a few slides on the data distribution. And here we have HGS-GET, which streams data from a location and supports range queries on indexed and BAM files and VCF files. The advantage of this is it means that a user doesn't have to download the complete file. So for a BAM file, you could be talking about 100 gigabytes of data. The user can just request the region or gene, for example, of interest that they wish to pull down. It's compatible with existing NGS software and pipelines, and it can support secure access via login using, for example, the ELIXIR AI and secure HTTPS distribution. The EGA Data API supports the GA4GH HGS-GET standard, and this data API underpins the EGA Python client and Fuse layer, which make accessing EGA data easier. So our data API, it creates a set of REST endpoints for data access, and it can list all the data that user has access to, plus allow the user to fetch this data. And it supports programmatic queries. And it's the support for programmatic queries that allow the use of the Python client, which is a command line interface, so to interact with the data API. So using this, you can actually list the data you've got. You can see the data sets, the files that you've got, and you can transfer the data down, or you can transfer regions of files to you. EGA is also using the GA4GH standard for data encryption. And this allows data to be encrypted by only encrypting the header with a specific key. The rest of the file is encrypted using a key which is encrypted in the header. This has significant advantages on distribution of data because it only means that the small portion of the file in the header needs to be re-encrypted when distributing the data. It also increases security because encryption at rest is supported by default. So using HGS-GET and the AAI, here's a use case for how these linked in together. So we work with RD Connect, who are based at CINECA in Barcelona, and they submit data to us, but they have their own genome-phenome analysis platform, which allows users to visualize BAM files and investigate the SNPs and variation within those. Here, a user can now log into the genome-phenome analysis platform, and they can pull up a region of a BAM file which is hosted in EGA, and that region gets sent to the GPAP, or Genome Phenome Analysis Platform, via HGS-GET for visualization with a genome browser. For example, here, this is the IGV browser. And you can see now that you're extending the access to data in EGA, which traditionally is behind this controlled access curtain and was hard to gain access to users who just have to log in, and then they can start using a browser to browse the files. So in conclusion, so the EGA with ELIXIR and GA4GH is developing the standards for interoperability, and these support the federation and ultimately extends the data access. The EGA is a driver project for the GA4GH, and ultimately the aim from all of this is to make the controlled access data more fair, which is findable, accessible, interoperable, and reusable. Thank you.
Excellent, thanks Dylan. I'm monitoring the open air, but there don't seem to be any questions. I do encourage all that are online to submit your questions in there, and I can relay them to Dylan. Or for those that are connected directly to the Zoom app, I can see there are currently 92. If you type your question into the window, I can relay it. But for now, Dylan, I have a quick question, which is, you talked about the geocodes. If you look at the data sets that are within the EGA, what's the number of data sets that are marked with these geocodes, maybe as a percentage?
I think it's around 650 now that are marked on that. So data sets that come in now and have been for, I think probably a year or so, have had to have geocodes being applied to them. But obviously there's the legacy data sets, which we are going through and trying to curate, but that is an ongoing process.
Excellent. And then related to that, if you look at the DACs that could use REMS using these geocodes, is it a similar process there, that the newer submissions are being encouraged that way to use the duo and REMS combination? Is this in production? Is that being used?
REMS will be able to work with developing this permissions API that I mentioned. Once that comes online, then that would allow REMS and DACs to be able to use that automatically. There's no other work that they would need to do, because obviously we're already using Duo for all the data sets that come in as of now anyway.
Excellent, thanks Dylan. I'll open the floor up to the co-chairs. Are there any questions for Dylan before we move on? It looks like Mikael's trying to talk, but I can't hear you, Mikael. You might be on mute. Okay. Okay, then thanks, Dylan. That's really great. Melanie, please share your screen and provide the next presentation.
Great. Hi, everybody. Share screen. PowerPoint. Here we go. Share, and please do let me know if anything goes wrong. All right, so hi everybody, it's my pleasure to be here today to tell you about my work for metadata standards, and specifically in the context of ELIXIR and the Global Alliance for Genomics and Health, how we're building that international coordination around standards and deploying them at scale. A quick reminder for those of you who may not be super familiar with GA4GH, the Global Alliance for Genomics and Health. Mission statement: we are trying to accelerate progress in genomic research, human health, and we want to build a common framework of standards and harmonized approach so that we can impact genomic data sharing and eventually health-related data sharing. So the ultimate goal is making an impact on human health. And when we try and look a little bit more in depth at what this means, you know, building that common framework has a basic number of steps. We need data models, which will allow us to represent both access condition of this genomic data set. Dylan was just talking a little bit about the data use ontology, and also being able to represent the cohort data, the clinical data consistently. We need tools and processes for implementing those standards. And finally, we need to be able to deploy those at scale across clinical cohorts and leverage them to enable related querying. So the first step, as I mentioned, is building data models. I wanted to talk a little bit about the data use ontology and also our latest product, the genomic cohort knowledge ontology. So the data use ontology, Dylan just showed you how it works over the EGA, the European Genome Film Archive. You can browse the ontology itself on the links I pointed there, the GitHub link. Very briefly, it's a list of terms and it's arranged in a hierarchy, and you can use those terms to tag your data set, and you can use those terms to search. Here's an example of how you can search using geocode on EGA. And you can use them to request data access. So as a researcher, I can come in, I can say, I'm interested in doing disease-specific research using that Mondo term, which I think is brain glioma, if I remember correctly. The advantage of using the data use ontology, it's version. You have different versions. The version here is the version from 2019. A new one has been released since then. And it's also machine readable. So if you keep in mind that idea of providing access at scale, obviously we want to try and decrease manual intervention, and any of the standards we're developing needs to be machine readable and relying on existing tools. So in this case, the Web Ontology language, which is a World Wide Web consortium formats recommendation. The other data models I wanted to talk about is the generic cohort knowledge ontology, or GECKO, which we've just released actually at the end of last month. So this is an outcome of the CINECA project, and I will tell you a little bit more about the CINECA project later on. It's again an ontology, so again a hierarchy, but this time we're trying to identify terms across cohorts that are common and that we can harmonize again. So if you consider, for CINECA, we've considered 9 cohorts. Here's a little example with only 3 of those. So if you consider those 3 cohorts that are all, they're all describing human population with genomics data set, some sort of assay, maybe, some sort of lifestyle and exposure. How many hours do you sleep a night? Have you ever been pregnant? Things like that. At the moment, those cohorts are very disparate in the way they represent each of those attributes, which are the black dots here. So what we are doing in CINECA is trying to understand from the technical implementation, what are users searching on, what's the type of query they're doing, what results are we returning, which cohort are bringing back results, and also from the use cases, what's the interesting query we can do against that data, what are people interested in doing. I'm interested in mental health, which of the cohorts have data on Alzheimer, and we're trying to analyze those together and have that kind of feedback loop to bring those together and hopefully lean towards the right-hand side of the diagram here and have that overlapping set of attributes which we are capturing in GECKO. So as I mentioned, this is early work. You can browse the ontology here. Contributions are invited. We've really done just a first initial release. If that's something you're interested in, I would love to hear from you. We have a GitHub, it's all public and open. Please do join us and contribute. The second step, as I mentioned, is having some sort of tools and processes that people can reliably use when they're trying to do validation and automated curation. And where that's important is, often metadata is a little bit of that broken telephone game where the initial scientist that provided the samples to the person who's entering the data 3 years later in the submission system. A lot of things have happened. The postdoc left, the format of submission changed, the submission interface suddenly require data that's actually not readily available. And it can be hard to really go back and forth and information get lost, and that's a problem. So some of the things that, some of an example where that's a problem, for example, for tool support, last month, I guess, end of August, my colleague at AGNC, the Gene Nomenclature Consortium, have just released an updated guideline. And sometimes it is easier to rewrite genetics than actually trying to update a tool as common as Excel, which a lot of us are using. And an analysis of papers was done, and almost 20% of papers, so that was an analysis of, I think, 3,000 or 5,000 papers. In almost 20% of them, the data is mangled because Excel renamed symbols. So sept Sept1 was renamed as September, common names were changed. It was just a big mess, and it turns out it's easier to actually rename genes than trying to update common tooling, and that's a problem when you're trying again to do things at scale and have reliable suites and workflow you can trust throughout. So my work at EBI, where I'm based, I lead the BioSamples database. Dylan mentioned it briefly earlier. We're trying really to make that metadata hub for the Institute, which we're mirroring, again, international effort. We're mirroring that with NCBI in the US and DDBG in Japan. And the goal of BioSample is to be really that gateway to other archives. So the metadata is stored centrally in BioSamples, and then you have link from each of those metadata points towards a different archive. You can recognize the symbol for EGA that Dylan just described here. We currently have over 12 million samples metadata. And one of the things we're really pushing for in BioSample is trying to make data fairer, so findable, accessible, interoperable, reusable, and do that in a consistent way that other people can also reuse. So our core mission, adding value between, on the left-hand side, people that provide data. We're working with a certain number of established projects, but anybody can submit data to BioSamples. We're trying to connect them to the right standard. We're trying to do recommendation of attributes, and I'll show you an example later. We're trying to link better towards external resources. We have more and more demands on domain-specific needs. So, for example, we're working closely with plant scientists at the moment. And we also have, on the other hand side, we have people that consume the data that, they want to be able to search. The Global Alliance for Genomics and Health wants to be able to retrieve all data sets of patients that are less than 50 years old and that have cancer. And we're really trying to make that search more relevant, more complete, and available, the data sets available for further integrated analysis down the line. The data I mentioned, this slide is a little bit old. It represents 10 million samples here. We're having a lot of data. It's a lot of data. But we have a lot of it, so we can actually try and leverage automated curation processes and try and clean it up and cluster it and see what happens. So we've done that. We've done, first a word cloud on do we have a problem in BioSample? And you can see, we have big ones. We have collection dates with 1L. That's a simple typo. Different versions of attributes are used. Some have underscores, some don't. The cases can be different. And for example, the collection date happened in nearly 1 million of cases. So you can really see that that's clearly a submission pipeline, and that's really amplifying mistakes and propagating those mistakes through that broken telephone line towards the downstream users. So what we're trying to do, we've looked at how the distribution of attribute is done in BioSample, and we can see that a very minimal subset of attributes actually cover pretty much the whole spectrum of data in BioSamples, and we're trying to focus on those attributes as primary targets for automated curation. As I mentioned, we're trying to cluster and see how attributes are similar to each other. So unsurprisingly, latitude and longitude, the big black blob in the middle, are very close and similar to each other when you cluster samples. You can also see the orange and green clouds, which are both human metagenomic data. They're just slightly different, and they use slightly different standards, but you can see that they're still relatively close. So maybe, that's an indication. Should we try and have a common standard for human metagenomic data? Is there something maybe already that people are not aware of? Could we recommend when, if the orange data pre-existed, could we recommend when the green data was submitted? Could we say, have you considered renaming your attribute that way? So those are all tracks we're exploring at the moment in BioSamples. The second point I mentioned with respect to tooling is about validation. We had a validation implementation study, which I co-led with Frederick Coppens, and we've deployed a common ELIXIR JSON schema validator as a node library, so anybody can go there, reuse it, deploy it. Locally, we've used it for the human cell atlas, which is a GA4GH driver project, and we're using it very commonly in BioSamples, which is an ELIXIR deposition database. An example of how we're using it in BioSamples is towards building that recommendation engine I talked to you about a little bit earlier, where you have samples currently, and you can see towards the bottom of your screen. We just released this. This is still the development version. So we have certificates on samples where we can say this sample complies with BioSamples minimal. We just require you to have an organism. And also this sample complies with an NCBI candidate schema, which actually is a schema that will guide a plan for curation. If you look in that sample, the attributes INDC status is live. The official recommendation for INDC status say this should be public. And at the moment, we have that mix of live and public, which is really painful where you're trying to search across samples. And so what that certificate, the last certificate here does is say, you know, you could curate that sample automatically if you wanted to, and you could curate it in such a way that it would be compliant with INDC status by updating the value to public. So this is really a, you know, a basic first example, but this could really be leveraged at scale again. When people submit data, we can recognize the type of data it's submitted. We can say, hey, we've noticed you've entered collection data with a typo, let's correct it. We noticed you're entering human metagenome data. Have you considered filling in that information? So we think this is very promising, and I'd love again to hear from you if you have any suggestion on that. The last point is, you know, bringing it together, taking those standards, taking those tools, deploying it over clinical cohorts, and trying to do federated querying, bringing genomics data sets together. So we've done that in CINECA, and as I said, CINECA is a common infrastructure for national cohorts in Europe, Canada, and Africa. You can see that it's a very disparate set of cohorts with different type of targets. Mamana later on will talk a little bit about H3 Africa. We cover disease and population. We have longitudinal studies, and we're really trying to bring those 10 cohorts over 3 continents, 1.4 million participants together, and do those queries across those larger numbers of people. And in CINECA, what we've built, we've used the genomics cohort knowledge ontology, GECKO. We've built a pipeline that takes the CINECA cohorts, the existing data set on the left-hand side, harmonizes the metadata across those cohorts, build a consistent representation and view over those cohorts, which can then be leveraged by GA4GH standard, for example, Beacon or the search API, and make that available either for downstream users in CINECA that have clinical use cases they want to ask the data about, or even potentially down the line external users. The next step for this work is implementing authorization and authentication, as Dylan just described, using AI and the data use ontology. So this is what we're currently working on. The last example I wanted to talk about today is the International 100 Cohort Consortium, IHCC. Similar goal, create a global network to address those scientific questions that, you know, if you look only at one cohort, there's no way, you don't have enough data point to answer it. But if you're looking globally, maybe you can start looking into rare disease, and I think we'll have a presentation from Núria later on. IHCC is interested in cohorts that have over 100,000 participants. In the last census, end of 2019, they had 61 cohorts, that's 30 million sample across 32 countries. So they're really casting a wide net. And the goal is, again, combining those cohorts with translational research and to impact human health. So what we've done here, we've taken those cohort data dictionary, which are, as I told you earlier, very, very disparate, and you know, everybody's using their own favorite vocabulary. And we're trying to bring those together. The way we're doing that is building a consistent representation. And this is available for a registry application and the link on your screen. And we're, again, thinking about versioning. How can we detect change? How can we enable cohort owners to submit their own cohort dictionary? How can we do those automated curation and mapping so that we can propose mapping to user and really make it as easy as possible for new users to come in and reuse our system. And the way this has been used is in the IHCC cohort atlas. where we've taken the IHCC cohort and we've done that mapping and harmonization work on dictionary on a subset of them. And we've rendered those in the IHCC Atlas, where the focus is really on discovery. So it's really about, I want to find cohorts that have data about blood pressure, or I want to find cohorts that have data on mental health. And so you can do those queries through faceted search in the IHCC browser. This is still under development, so this is really a sneak preview just for you. This is an internal, but should be released very soon. In summary, so we are trying to bring together those data models, tools and process, and deploy those over cohorts such that as a researcher, when you deposit data, so that's the top arc, when you're depositing data, you can validate those data. You can benefit from recommendation from your deposition pipeline. Curation is applied automatically down the line for you. So we do tagging for ontology term. We do the automated curation that I showed you earlier. And that data gets stored in BioSamples. And through control access, you can get access to the data sets from EGA. So some of the CINECA cohort, for example, the data sets are in EGA. On the other hand side, if you're looking at the bottom of the picture as a researcher, you can then consistently find the data. So you can use a system like Beacon. Do you have a genome with a specific allele at a specific position? Or you can use phenopackets and schema block. And I think Mikhail will describe those a little bit later to query by phenotype if you're interested in specific patient phenotype. So this is really about building that ecosystem for cohort federated querying. So that's all I wanted to talk to you about today. I'm obviously not alone working on this. There's big groups, so GA4GH, clinical and Phenotypic workstream, the data use researcher ID, the discovery workstream, the metadata standards team at NTB-EW02, they're amazing. We've done a lot of work with Dylan and EGA, and we've had a lot of support from ELIXIR on validation and the fondue implementation study for plant. I'm happy to answer any question.
Very good, thanks Melanie. It might take a second or two for the questions to come in, so please do submit your questions and I'll ask them to Melanie directly. As we wait for those questions to come in, Dylan, there was a question for your presentation from Venkata, asking whether the encryption of the header files requires special tools to decrypt, like VDB decrypt used for dbGaP data. Is that the case?
I'm not totally sure. I know that it's supported by SAM tools and so on, so I would suspect not. But what I can do is actually post a link to the specification in the chat if you want, and then you can look through themselves.
I think that'd be really helpful. If you post it in there on the open air window, then Venkata could read that and reach out to you if the answer doesn't forthcome. So are there any questions from the other co-chairs to Melanie on that presentation?
This is Kaisa from Finland. I would like to know, to ask Melanie about the IHCC cohort and how can you join into it if you have interesting cases that fit.
Sure, so the IHCC is a consortium, and I think the way to join would be reaching out to them, and I can put you in touch with the people, or you can reach out through their website. The one requirement to join IHCC is that your cohort need to have over 100,000 participants. So
Okay.
As long as you fulfill that requirement, you can totally join the IHCC as far as I know.
Yeah, okay, thank you.
You're very welcome.
Okay, that's really great. I don't see any more questions coming in for you, Melanie. Thanks a lot for your presentation, and I hope you can hang around and the rest of the workshop.
Will do. Thank you.
Excellent, great. For now, we'll move on to Mamana. I think you're the next presentation, so please do share your screen, and it's your option if you want to open your video or not.
Sure. Thanks, Gary. Just going to try to share my screen. Okay, can you see my screen?
Yes, that works well.
Okay, thanks. Hi, everyone, and thanks, Gary and Melanie and all the organizers for the invitation to come and talk about East Africa. So my name is Mamana Mbiyavanga. I work at the University of Cape Town in South Africa, in Cape Town precisely. And we're part of East Africa that I'll be talking about in this presentation. And my presentation will be about the impact of the Global Alliance standards on the East Africa infrastructure. So I'll quickly start by introducing East Africa, and then also discuss about the EGA, which is one of the projects of East Africa. Then quickly talk about the engagement that we have with the Global Alliance, then showcase some few use cases of how we trying to use Global Alliance standards. So East Africa is an initiative that was launched in 2008. And the aim was basically to try to facilitate or to enable genomic research in Africa, so that we can improve the health of African populations. And that was really trying to emphasize on a research done on Africa by African researchers on different diseases, common diseases and rare diseases that are pertinent to the African continent. And on the picture, we're seeing some of the pillars of East Africa. The first one being in an effort to build capacity to create centers and research projects that will be doing research on different topics of interest to Africa. And the second one is to generate data, and as we're generating data and collecting samples, to put in place repositories that will host the data and the samples that we are collecting. And the third one is really to try to enable or facilitate data sharing. As we've seen in previous presentations, how important that is. So as we're generating data in East Africa project, we're really trying to make sure that we're able to share the data that we're generating so that it can be useful for the whole community. The third pillar is while trying to build capacity is to try also to put in place sequencing centers or center that will be able to generate genomic data. And one important pillar that I have also to mention is training and mentoring. As I said, while trying to facilitate or to enable research, genomic research on the African continent, we really want to make sure that human capacity is built so that research or genomic research or any research that is done in East Africa can be really be performed by African researchers. And the last one is, while we're trying to do all these things, is to make sure that we create collaboration within Africa. So that is by creating a collaborative network of African researchers that are working in genomics. So just some highlights in numbers of what EGA Africa is. There's about 176 million of investments that is now that has been invested in EGA Africa, and we on the second round. So the first round ended in 2013. Then we started the second round. There are about 500 members that are part of EGA Africa from different institutions. We have about 30 countries on the African continent that have project or research centers in EGA Africa. Now we have about 51 research projects that are supported. And one interesting thing that I have to mention is that we can see the number of trainees that are being trained as part of EGA Africa, PhD students, master's degree, the workshops that we've been able to organize, and so on. And we expected at the end of the second round to generate about 10,000 samples, and the recruitment is still going on. So on the map, we're seeing some of the sites of EGA Africa. As you can see, it's spread almost all around Africa, Western Africa, Eastern Africa, and Southern Africa. We need to make sure that the whole Africa is covered by genomic research that we're doing. And we're also seeing some of the diseases that we research project in EGA Africa working on, going from mental health, cardiovascular diseases, infectious diseases, and some other genetic disorders pertinent to Africa, including sickle cell, for instance. So the consortium, as I said, is made of collaborative centers. We have about 13. And the research project that we have of different types, going from research projects, completely research looking at diseases or phenotypes. We have training programs for mostly based in Western and Eastern Africa. We have established 3 biorepositories where all the samples generated as part of EGA Africa are stored, hosted, one in Western Africa, one in Eastern Africa, and another one in Southern Africa. We also have 11 projects or grants that are looking at ethics-related project. And all this, all of this is catalyzed or facilitated by a network of Pan-African institutions working in bioinformatics, which is called EGA, and which is trying really to develop bioinformatics and data analysis skills, and also trying to build infrastructure for data transfer, infrastructure for analyzing data that include compute, tools, workflows, and also looking at data management from collecting your samples until generating your genomic of the data that you're going to use in your analysis. So how do you manage that? Also trying to put in place tools that you can use to harmonize data across different projects. BioNet has 28 partners from 17 countries in Africa, and we have about 120 members. So as I said, ELIXIR focuses on data management and standards. That's why we're part of GA4GH, because we're interested in using standards that will allow or facilitate sharing of data. We're also looking at pipelines and computing, tools and web services, data integration, databases and resources, and also making sure that whatever we're doing is sustainable and communicated through outreach to appropriate stakeholders. And all of this is related to training, so that everything that we do is communicated, or the skills is transferred to ELIXIR Africa consortium. So ELIXIR Africa is part of GA4GH as a driver project. So we take part into some work streams. We're also part of the steering committee as driver project champion, and we're also helping co-lead a subcommittee in GA4GH. And we're also part of the GA4GH partner engagements, such as the Genomic Health Implementation Forum, where GA4GH tried to put in place environment so that partners or driver project can engage or communicate. So this is a summary of some of the Global Alliance standards that we basically either adopting or contributing to. I think Dylan took us through many of them, so I'll not take time on this slide. Just to mention on red are the standards that we currently adopting or contributing to. That includes Phenotypic, pedigree representation. In genomic knowledge standards, we are looking at variation representation. How do you represent variants? In data use and research identities, we are looking at DUO and also GA4GH passports. In discovery, we're interested in Beacon and also service info and registry APIs. In large-scale genomics, we're interested in rich file formats that includes SAM, BAM, CRAM. I think Dylan mentioned CRAM. I'll also come back to that. We're also looking at VCF and BCF file formats, and also crypt for GH. So just to give us quickly an idea of the context, how we do research in ELIXIR Africa. So starting from recruiting your participants. So we collect or we generate 3 kind of data. The biospecimens that go to the biorepositories. We're also collecting clinical data and then generating genomic data. And for clinical data and genomic data, there's data management process that goes in there. How do you manage that and so on, but also data analysis, of course, to try to generate knowledge for that. And in ELIXIR Africa, because we don't have any facility or infrastructure for long-term storage of the genomic data, all the data generated in ELIXIR Africa are sent to EGA and INEA for long-term storage. So going further, we work. So once a participant is recruited, as I said, the biospecimen will go to the biorepository, and clinical data will go at the end with the genomic data into EGA, There we try to map ethnicity and demographic data. As you know, that is very important to Africa because of the number of different ethnic groups that we have on the African continent. We also try to map CRFs to ontologies because different projects have different CRFs that they use to collect the data. We also try to map to data ontology. They're using the GA4GH DO, and also try to harmonize where is needed. And also we have what we call the East Africa participant recruitment database, where we store or we track the progress of participant recruitment. Coming to genomic data, we are using CRAM formats and VCR formats. And because of the different projects or countries that's involved in East Africa, we're looking at Beacon so that we can have federated, kind of sharing data where each country or each project can share the data. And we have what we call East Africa Archive, where all the data that are generated in East Africa before going to EGA are stored. There we're looking at using HTS gates, CRAM and VCR formats, also looking at using GA4GH and also passport for authentication. We're also developing what we're calling AGVD, which is the African Genome Variation Database. There we're looking at using CRAM, VCR formats. We're looking at also using the variant representation and variant annotation, and of course, passport for authentication. And there, you can see the AGVD will also feed the data to the African microbiome portal, the African Genomic Medicine portal, and also to the African, the East Africa Imputation Service that we busy deploying. And once the data is in the East Africa archive, we also busy building a catalog where people will be able to search and request data from East Africa. And all the data that will be in the archive will also be provided to the community through Beacon. And also the data that will be submitted to EGA will also be provided for sharing through EGA Beacon. So to finish, just a quick, some few use cases. So we're using DUO to map all the East Africa data for restriction and for appropriate usage. So once the data is generated in East Africa, there's a 23-month embargo on the data that gives the researcher or the data generator, the project that generated the data, the opportunity to work on the data and to be able to publish any result before the data is made available to the whole community. So it's in the nine-month embargo that's issuable nets. We take the data that is submitted to us, then try to map that to the DUO term so that it has appropriate authorization or restriction for use. So far we have mapped 20 East Africa projects, of which 8 are in EGA. And we had some interesting interaction with the DUO team to try to work on how we can improve or contribute to DUO, making some recommendations so that DUO can be useful for East Africa. The second use case is the CRAM format. As I mentioned, that we were thinking or we're using that for in our ecosystem. And we now all the data that we're storing for long-term are converted into CRAM. So far, we have about 8,000 human exomes and genomes in CRAM, which are in size about 107 terabytes, which in BAMs could be about 600 terabytes. As you can see, we're gaining or we're saving about more than 50% of our storage. And we're also looking at GA4GH for data access and sharing in the data archive, but also when we exchange data or send data to EGA, we're looking at how we can use GA4GH to try to encrypt data, taking advantage of all the benefit that GA4GH offers. I think that's all, and we funded by the NIH and Wellcome Trust, as I said, and also the African Academy of Science. And thank you. Happy to have any questions if there are any. Thanks.
That's really great. Thank you, Mamana, for the presentation. On the OpenAir app, I don't see any questions coming in, but there was one from Tommy on the Zoom chat. Tommy, would you like to ask your question directly?
Sure. So thank you, Mamana. I think this is an excellent presentation. So my question was about adaptation of the GA4GH passport standard for the data access. So can you share any experience that you have had, or is it just a plan yet? So how do you express the data access rights in the H3 Africa ecosystem?
So thanks, Tommy, for the question. So I'll say that's the plan that we have to extensively use the GA4GH passport. But so far we've not yet implemented any of this in our ecosystem. But there's a plan. It's a plan that we're looking forward to implementing. But for some of the service that we're deploying now, where we really need to have a proper AI in place, we're planning to use the ELIXIR AI until we're able to deploy our own AI.
Great, thanks.
Okay, are there any other questions that I've missed that the co-panelists have seen that could be asked?
There's a question on the OpenAir, Gary.
Can you see it, Melanie?
Yeah,
Okay.
So it's a question from Núria Queralt Rosinach about, I wonder how do H3 manage integrating ethno-linguistic ontologies with other commonly used GA4GH ontologies?
So when we started H3, we tried to look at ontologies or ethno-linguistic ontologies that exist, and to see how we can use them. But then we realized that most of them were not really useful or complete in the sense that they're not really including all the ethnic groups that we have in Africa. So as part of H3, we started a project where we're building our own ethno-linguistic ontology. That's most of the H3 project are using. So in the, I mentioned that we also interested in using the pedigree representation that GA4GH is building. So there we're trying also to try to see how we can use the ethno-linguistic ontology that we're building so that it can be useful to the whole community. So we're using the ontology that we're building in East Africa for that.
Okay, thanks, Mamana. There are one or two more questions coming in on OpenAir, but we're running a little over time, so I would ask you to use that platform and answer those questions directly. We'll move on in the agenda for now. And
Great,
the next,
thanks. Thank you.
thanks, Mamana. The next presentation is from Núria, and this covers the rare diseases use case. And I have a specific thanks to say to Núria, who stepped in right at the last minute to give this presentation only yesterday due to some holiday and everything else. So Núria, thanks a lot. And I ask you to share your screen and deliver your presentation, please.
Thank you very much, Gary. Yes, doing this presentation on behalf of Dr. Michael Ruth, and I will present this the rare diseases from the perspective of the European Joint Program on Rare Diseases, how we are applying FAIR principles via semantic rigor to practical use of rare disease resources for more effective research. Can you hear me properly, by the way?
We can hear you well. For your, would you like me to share?
Yes, please.
Okay, I shall do that now.
Yeah, sorry. So I will be presenting all about rare diseases and the virtual platform that we are working on in order to provide a federated platform to enable more sustainable and effective research for the rare diseases. I'm very sorry, I'm a little bit nervous because I couldn't prepare properly the presentation, so I'm very sorry. And also I joined the biosemantics group pretty recent, so I'm a newbie on the project, so I hope that I can answer all your questions.
I'm sure you will. So I'm now sharing my screen, Núria. So if you just ask me for the slide, we can present the presentation.
Okay, so we can go next to the next slide. So yes, a brief outline of what I'm going to talk about is, first, I'm going to present the rare disease research problematic barriers and approach that the European project is having. Then I'm going to present what is this the EGPRD project. Then I'm going to present the overlap of this project with the suite of tools from the Global Alliance for Genomics and Health. That is mainly that we are providing a driver use case for Global Alliance. Also, I'm going to discuss about, because the main approach of the project is using semantics, it's a semantic approach. So I'm going to talk about all about semantics and the virtual platform. And finally, also, I want to explain the overlap that the project has with ELIXIR. That is basically that ELIXIR is helping to realize making it real, the virtual platform. So next, please. So rare diseases are those diseases that have a very low prevalence, and there are several definitions. In Europe, a rare disease is defined like a disease that affects 1 person every 2,000. But globally, there are more than 7,000 rare diseases. So it's a very big problem, not only for patients, obviously, that typically patients are suffering for very severe symptoms and conditions during early years in the childhood, or from they develop the disease as well. If it's in a more adult time. But also healthcare systems and providers are suffering a lot in order to try to provide an optimum care for these patients, and also for the society that economical burden. So the problem is that these patients are so rare, these diseases are so rare, there are so many few patients that they usually have a very struggle process from when they are diagnosed. That is not easy at all. Can pass like 7 years until they can reach, if they can, an optimum treatment. So this is called like the diagnosis patient journey. So in order to improve this, to improve patient, low the suffering of these patients and improve the lifestyle, we need efficient research. But the problems, the barriers for an efficient research are several, mainly that the expertise is very sparse and scattered. Maybe there is an expert on your rare disease, but it's geographically very far away and difficult to find. Other barriers are the lack of data, because there are very few patients, the lack of knowledge about the pathology, pathological mechanisms underlying those diseases, and also lack of cures. So the main goal, the very big goal of the European project is to enable these patients to receive an accurate diagnosis as soon as possible, an optimum care, and provide therapy within the 1 year of coming to a medical attention. This is like a very big goal. So the strategy of the project is mainly based on enable coordination and sharing, but at all levels, not only for research resources, that also, or infrastructure, but also like a hub for all the stakeholders to be able to coordinate, align, support, share knowledge, expertise, legal issues, whatever. So next, please. So the EGPRD project is a very big project. It brings over 130 institutions, including all these 24 European reference networks. That these networks are basically networks of healthcare providers that concentrate expertise, knowledge, is a hub to discuss about possible diagnosis or treatments or even resources. Yeah. So it encompasses like from 35 countries, 26 in Europe, and 7 associated in UK and Canada. So the main goal is to create a comprehensive, sustainable ecosystem to circle between all these efficient research, efficient care, and medical innovation. Next, please. So it's a big project at organizational level. Is divided by 4 different pillars. The first pillar is basically enabling collaboration for funding, to find funding and resources. The second pillar that I'm going to focus on this presentation, that we are working is embedded in this second pillar, is mainly for research, to develop this virtual platform for data and resources. And the approach is to use to apply the FAIR principles in order to make an efficient, reliable, and sustainable research. Then there is this pillar 3 that it entails training and dissemination and all these alignment and coordination between patient advocacy groups, clinicians, researchers, healthcare providers, whatever. And finally, the last pillar is focused on translational processes. So it tries to enable to accelerate the process of the research outcome, from the research outcome to an efficient drug development and quickly find new treatments and all. So next, please. So here it's mainly the main idea of what the project is wants to provide as pillar 2. We want to provide this virtual platform, this federated platform that mainly the idea is to query across different kinds of resources to enable analysis in a secure infrastructure to keep data privacy, but we want to connect all these resources that you can see are very diverse. Types of data are very diverse, catalogs of registries, biobanks, different kinds of tools for analysis, different kind of vocabularies, different kind of models, so different kind of terminology, different kind of data schemas. So we want to connect this, but without moving data from the source. So we want to query all these sources in a federated way. So you can imagine that this is a challenging problem to try to connect all these together. And also, we are working with Reddit experts in these European reference networks to provide support to verify data about health care and patient registries, and yet to provide this fair data at source. So the idea is that the data is not going to move from the source, but the analysis is going to move. It's going to visit the data. So next, please. So the approach is to develop this virtual platform of data and services for analysis and efficient research, to connect all these disparate and heterogeneous resources and connect it and harness it in a federated way, and to provide support for all these European reference networks for to make this rare data fair data, convert this data to fair data. So next, please. So as I mentioned before, there are 24 different networks. Every network is specific to a disease group, like bone disorders, neurological diseases, metabolic disorders, and etc. And as you can imagine, here to verify data, it means also is a challenge to every network. Every network is going to have its own model and way to structure the data. So we are going to support this verification through data stewardship. So next, please. But as I said, this is a challenging problem. And in order to enable interoperability, we need to use standards that are used in general for the rare disease community. So we are working with the Global Alliance for Genomics and Health as a driver project. And because of the efforts put forth by Global Alliance to standardize and for genomic and health data to share across the institutions and founding professions, all this effort is very big and it's community-wide. So we are collaborating with this driving use case, actively contributing with the Global Alliance in different work streams, like developing ethical and regulatory guidelines and regulations, and also in the clinical phenotype data capture to develop, if needed, new standards for the rare disease community, or reuse the standards that are already in place, and we don't want to reinvent the wheel. If something is working and is used for the community, we are using it. So next, please. So one of the key goals is to federate, to do this federation, do these queries at resource level. So making, producing questions, firing questions connect to the disparate sources and across resources. So we have developed, or we are developing an ontological metadata model in order to cross all these resources. Oh, I think there is a problem with the graphic. It's not properly shown. It lacks a lot of boxes, actually. Well, mainly the idea is that the first year, the project was connecting different kind of data resources and biobanks and registries and tools. And from the second year onwards, we are expanding this with other kind of tools and resources. So the idea here is to search catalogs, make queries at reference level through the metadata. So next, please. So then also there is the access to the actual data. So and the other kind of queries that the project. Sorry, next, please. So this was a prior one. Okay, this is yes. Sorry about that. So the second type of query analysis that we want to provide with this platform is to federate, to fire a question, go to the sources, to the disparate sources, and query across the data. So that means that we need this data to be really interoperable at the record level. So this requires the use of common community standards. Also to work a multi-tier software development to enable this fair ecosystem to go to the sources, because the project is only centralizing the virtual platform, the portal to do the query, but the query is going to go in a distributed way to the source. And finally, for all this happen, we need to combine expertise for doing this verification support. So from software developers to data stewardship and also AI experts for enable access in a controlled way for data privacy issues. Yeah, so next, please. So to access all the file queries and access it at level of research, as I said, we are doing this through an ontological metadata model. So to do that, we need this verification process to happen at source. But this doesn't mean that we are forcing one model and one vocabulary for all. So, but of course, because these resources have to interpret and have to be machine readable, this requires some kind of harmonization among these certified resources. So the solution that or the approach that the project is taking is at least to provide a canonical data model to transform the main data elements, and also a canonical metadata model to transform this metadata model. And all these, the approach is to do this in a very semantic way and rigorous way. So next, please. So for a level of data, a lot of effort right now is in progress, is in providing this semantic modeling of the common data elements. These common data elements were for patient registries were provided by the European Commission Joint Research Center. So we are basing on these common elements and applying semantics and applying semantic modeling in order to make this canonical machine data model. So next, please. So as I said, we are enabling all this interoperability, the approach is using semantics in the virtual platform. What I mean for semantics is we are using semantic web technologies. So for describing the data in a structured way, we are using RDF and related languages like OWL or checks and shackles for model validation. Also, we are using triple stores and Spark endpoints as the infrastructure for query these RDF data. Regarding the metadata, we are using this vocabulary, the GECKO, and also APIs, developing APIs for machine-readable metadata at source through the Federated data point specifications that it's been developed under the EGP. So also the approach is to maximize the reuse of existing vocabularies, again with ontologies. We don't want to reinvent the wheel. Also, we want to use this called cool URIs in order to make identifiers persistent and they're referenceable always. And also, we are using or reusing already established models. In this case, the Phenotypic model from Global Aliens for genomics and health. And we are developing the semantic version of the Phenotypic model, being able to provide this semantic model as source for federated analysis. So without not moving data and Phenotypics from this. So next, please. So the consequences of this approach regarding the data is that it requires a huge investment in model construction. It's time demanding, is a lot of effort. Also, there is the problem that there is a need of tooling. We need tools to find resources, specific resources for rare diseases, also tools for aligned models, different data schemas, and also mapping, ontology mapping resources. These kind of tools, this is a very big problem. Also, another problem is that we need a kind of convergence and harmonization of these different models used for the European reference networks. And also there is a lack of a generic framework to transform this data. But the positive things is that the semantic approach, the use of ontologies and very well and structured ontologies and logic ontologies, formal logic ontologies, is, for example, to harness these semantics, these structured semantics with the inference, logical inference, and also this approach enables federation. So algorithms are visiting, querying at source, but data is not moving. So, and as I said, we are based. Our base is the semantic web technology. So next, please. So in order to help us to provide tools and resources, we are joining forces with ELIXIR. And they are supporting the effort for verification, for the verification process and the specific needs for the rare disease community, but also to use already guidelines and standards that are in ELIXIR. So it's a virtuous cycle, it's a win-to-win relationship again. We are providing the use case, and also we want one of the goals is to enable the framework for verification for the rare disease data. Also to enable within ELIXIR to develop these fair metrics for fairness evaluation, but specifically for the rare disease needs. So ELIXIR is providing infrastructure in a secure environment through mainly the ELIXIR interoperability platform. And also it is providing tools, resources for data analysis. So next, please. So a little bit to finish the presentation, some main conclusions is that we are developing this virtual platform the only thing that is centralized is the query, the portal that to query the data, but the data is disparated and is absorbed. So the concept is clear. The semantics is a core approach, and it's a work in progress, and it's a challenging progress, but it requires these joining efforts with this community initiative like ELIXIR and Global Alliance for Genomics and Health. So last but not least, my acknowledgements go for the Global Alliance for Genomics and Health, ELIXIR, and GO-FAIR for rare diseases, the Biosemantics Group in the Leiden University Medical Center, and of course the ICCB 2020 organizers for giving us the opportunity to present these projects and platform. So if I'm very happy, thank you again. I'm very happy to if anybody has questions.
Thanks, Núria. That was a really nice overview of the EGA-PRD. And I think there are some real key questions there about how you take the GA4GH products to the ERNs. So you go from technically minded population to clinically minded population. But I think in the interest of time, I'll try and take that discussion to the OpenAIR platform. We're running a little bit behind. So if you do have access to OpenAIR, I'll ask a couple of questions and there might be some others there as well. And we can follow up that discussion during the next presentation and the remainder of the workshop. But as I say, we're running a few minutes behind, so I would like to move on. So thanks, Núria, for joining, and I'll talk to you on the OpenAIR platform. The next presentation is the second use case, which focuses on cancer data. And this is from Michael Baudis. Michael, please share your screen when you're ready.
Okay, so you see now the presentation display?
Yes.
Okay, good. So let's go back. So I want to present today a bit of something different. It's not a national initiative or some theoretical groundwork we do to share data, although this will all be mentioned, but I actually want to use the opportunity to talk a bit more about how our own resource we have been providing for quite some time is moving and co-opting these standards developed through ELIXIR and for the Global Alliance, and how this is could be an example also for making cancer data and other data types more available. So the area we are interested in from the genetic and research perspective are copy number variants, and especially copy number variants in cancer. So copy number variants are duplications or deletions of genomic regions, sometimes in very complex formats, sometimes large scale, sometimes really involving only certain genes. And you see some examples here on the slide, like there in a glioblastoma, a homozygous deletion of the CDKN2A locus, but as part of larger rearrangements, or chromosome 13 duplications in colorectal carcinoma. Here the data is all from genomic array experiments, where then the signals from multiple measurements are segmented to derive these copy number changes. It's basically the same if you use sequencing data or array data. Exome sequencing data is not so good. Whole genomes would be good. Arrays are actually really good still. Now, how frequently do we observe this data in cancer? And this is a bit, to understand this plot, one has to think about how much of a genome is involved in any given cancer. And if you take each sample and look how much of its genome is duplicated or deleted and put this in, calculate this as a fraction, and then you plot this for these different tumor types, and then you can see here that up to 15 to 20% of cancer genomes are on average, depending on your sampling, duplicated or deleted. So have these copy number variant regions, which may sometimes be bystander effects, but also point to, and we know this, that there are strong selective forces on this. So lots of these regions are really involved in oncogenesis. Sometimes they are just the expression of genomic instability, but then even those have a selective pressure. So there's pretty much of genome variant, which leads into gene dosage phenomena, different expression of the involved genes genes coming from this region and so on. And you see the big differences, especially hematopoietic malignancies have fewer mostly of these types of rearrangements, whereas many solid tumors have a large amount of these changes, right? And you see there at the bottom right, you see example, chronic myelogenous leukemias, CNVs are nearly absent, right? We know BCR-ABL, main driver, and then some edit mutations, whereas in melanomas, you have many regions which are like chromosome 7 is duplicated in more than half the cases, and then also focal regions like I just showed the CDKN2A locus, thought was in glioma, now in melanoma it's too frequent. These changes can occur together and have a compound effect, like here, P10P1 and TP53 deletions in prostate carcinoma, and this has been known for a long time, that if these two changes occur together, then it's a very aggressive type of cancer. So it's good to look for these patterns and to understand co-occurrences of these type of mutations. This is an example now how data looks, and this is all data from our databases and from the online plotting tools. You can regenerate the stuff yourself if you access our resource. I will come to that later. Here, for instance, is now data from about 2,000 medulloblastomas shown, and you see the summary CNV profile, which gives you on the left side the frequency just of these changes in all these tumors, or how frequent are they. But if you look at on the right side, you see if you do the single sample profiles, yellow means always a duplicated regions, blue means a deleted region, then you see different clusters of genomic changes coming up. And we know now from many other studies that these genomic fingerprints of these tumor subtypes correspond to physiological subtypes, to pathophysiological subtypes, to different involved pathways in these different clusters. So they are actually, these tumors, although they look histopathologically the same, are different biological entities, just occurring in the same organ, but have to be treated partially differently and have to be considered as something really biologically different. And it's important to know that, and really to get these patterns and to understand this, you need lots of data. So this is I started now nearly 20 years ago, actually. This is something from 2001, the first version, or about the first version of the website. I've seen a poster from the first Stanford biocomputing meeting in 2001. You see that is this website at the top left, and accompanying publication then. But this has been then carried on then by me and my group then over the last decades, and many contributors who help us with the data. That's the current status of the site. It's actually not the current status, it's the not publicly totally linked version. It's next.progenetics.org, which shows a development version which just is upcoming. It is online, but the main site points still to another version. And you see there are now 113,000 genome profiles. About 90,000 of those are from tumors. The others are from reference experiments, because if you start to collect the data, then you also start somehow to co-collect very stuff which you wasn't interested in the beginning, but then it becomes actually interesting because now you have lots of germline copy number tracks too, which we also start to explore. All data is CCBY, so you can download and whatever with it, right? Now, such a resource is not enough, right? I mean, we put lots of work into that, and it's continuously growing. This will grow this year again with about 20,000 samples. But the problem is that as came up several times already, the current concept is more to look at federated data, because no resource by itself can deliver enough data for many research projects. And even if we have hundreds of different tumor types in our resources, we know we have many tumor types where we have just few samples and so on. And this cannot be grown alone by a single resource, even if you parse over other available data, which we take from publications, from GEO, from AreaXpress, from whatever resource. So Global Alliance has been mentioned many times already. Maybe this slide has been showed or something similar. Global Alliance looks at how to make genomic and health-related data accessible to users all over the world using sets of standardized tools, APIs, and so on. So for not necessarily for data sharing, but also for explorative analysis. So using different paradigms to make use of data which may reside in resources and different places. And the most successful so far practical implementation of a data sharing protocol has been the Beacon protocol, which was originally invented basically at the start of GA4GH itself, and the first implementation was done by DNA Stack, Mark Fiume, and others. And but this has now gone a long way. So originally Beacon, and this was mentioned now in several talks already, is something where you can send queries to a genome resource and it answers you, oh, do I have this variant in the data set or don't I have it? However, the Beacon protocol has changed quite a lot in the last years, and I will show you how we apply it to our resources. Driver of these changes since 2016 has been ELIXIR. So the ELIXIR Beacon project is a driver project on the GA4GH roadmap. So Beacon protocol is a GA4GH thing, but the project is, as the ELIXIR Beacon project develops the code, and I will mention later then the leadership and so on. So the version 1.1, which is still the currently active version, has seen lots of changes compared to the original protocol. So we have now structural variants, duplication, deletions can be queried. Others are partially possible as if the backends support them. There's a data handover concept, I will talk about this, which can be used to deliver data outside the original Beacon protocol. So you kind of keep the Beacon site safe, but can then use it for clinical and other purposes. And we see have already implementations, although they are not part of this specification yet, but have been defined rather precisely already to use filters for metadata to enrich queries and also to have extended quantitative responses. So the Beacon version 2 API will basically allow you something like that. And Dylan had shown this earlier already, this slide basically to allow you these complex queries where you have something like data use annotations, phenotypic annotations, genotypic queries, and these can all be sent to a network and then distributed. And then you're not able only to get a yes/no answer from these federated query, but may have many other options to make use of the data found through these types of Beacon queries. So it goes to a Genomics API with general use applications. Currently, what's possible, precise variants, like the first one listed here, have been always been accessible from the beginning. We have now range queries, basically saying, oh, you have a region in the genome. Do you have variants somewhere in this region? Variance frequencies are delivered, structural genome variants, and data matching. A query can then lead to a URL, which then allows you to use a different protocol to retrieve data if this is implemented by the backend and if you have the permissions for that. Important now for the upcoming thing, which are the filter queries. Dylan mentioned this already about implementation of this in the version 2, is that we can use now, we target these filters queries, as we call them. They are sent against ontology terms, basically using queries as the standard way to represent disease codes or other technical codes, experimental related codes, and so on. Duo, for instance, also is represented as a query. The interesting point, what we are now doing now in this council use case, is that we have coded all our data from originally ICDO, which we had coded also ourselves, towards NCIT, Neoplasm Core, which is kind of an emerging deep hierarchical classification of most relevant cancer entities. So which allows you then to do these, if you implement this in your backend, probably, then you can do hierarchical query selection, that you select any term on any level and then get all the child terms, for instance, for these matches. And this is very important for the type of Beacon version 2 queries we envision. So you can circumvent the problem with granularity or the implement indirect OR matching by making use of these hierarchical queries. For instance, you can query samples for colorectal cancer or specifically rectal mucinous adenocarcinoma, or combine these things. Yeah. So, and there are tools to do that. This is an example, for instance, showing just how it looks in our current website. You can there out in the hierarchy, you can open this up and so on. And if you select these two types of codes, then you see already below the first selection, you have all the child codes of these codes which exist in our database. And then you get all the response which will you deliver samples from all the child codes. And this is basically just using the current beacon plus already implemented filter query from version 2. So kind of fuzzy queries are enabled through that. And if you combine this with a data handover, you can do much more. This is a slide which you also can find on the Beacon project website. It's basically showing the general concept of what this handover means. You have a beacon query. The beacon query goes into your resource, and there is no exchange of private data, of patient data, of whatever. Resource handles their internals and then gives a beacon response, and just the beacon response, depending on which version and so on and how you shape it, can be just, yes, we have this variant. Now, it can also give you back an handover ID, which is as a URL or an ID pointing towards a resource access. And this can then be handled through a separate authentication system. And how your internal system handles this is up to you, but the internal system should then be able to have a temporary store of the data which had been matched. And if you're authenticated, you can use this URI to retrieve then the data. And this can be in any formats which you can specify in the handover object, which you return with your original beacon response. Could be phenopackets, could be VCF, could be graphics, and so on. So this is all already possible as part of the V1. And this is what we did. So I said we have a resource where we have more than 100,000 of these CNV tracks and lots of annotated metadata, which we map to classification systems and so on. And this would be now the beacon front end for that. And below that would be a beacon response for the CDK N2A query against more than 2,000 glioblastoma samples. So using glioblastoma NCIT code and using CNV query, which says, give me all the focal deletions which are smallish and hit this gene and in glioblastomas. And below you already see then the asynchronously retrieved histogram of all the data just using beacon query, giving a handover back, and then doing asynchronous query against the handover and delivering then the plot and the biosamples and so on, retrieved from this query. Then you can use just these objects, these handover objects, to go to other visualization systems. Says, okay, now with this handed over data, let me do some other stuff. And this depends then on how you implement it. In our resource, it allows you, for instance, to say, okay, now there were several NCIT codes in the query before. Now this is a different one, this one for skin tumors. And now show me the response split for all these different NCIT codes just from the samples matching the original beacon query. Now, going beyond that, and generally for resources, this was just a demonstration saying, okay, you can use beacon and a beacon-derived protocol with edit enhancement, doing maybe some other query types or so. But this already can power quite of a very informative resource, in our opinion at least. But you can also use many other data elements for different purposes. And as I mentioned before, multiple initiatives internationally work on kind of defining these standards and defining data annotations and data annotation systems. And the ELIXIR is involved, the EBI, as Melanie showcased, is very heavily involved. Lots of these standards are defined in the context of GA4GH. The Monarch initiative is in close collaboration and part of GA4GH and so on. And resource providers picking these standards up. And here are some examples of these standardized and kind of accepted formats like dual codes, like now experimental factor ontology, where we felt the need that there has to be an annotation to define samples and neoplastic. This exists now, so you can use the standard from these reference resources. Other standards could be GeoJSON or ISO codes for geographic location. This may be interesting to look for where your samples can come from, associated with environmental or population-specific factors. So all our samples and publications are, for instance, annotated using GeoJSON. You can search and scope them by region. Other things like always, how do you define an age? If you use a numeric term, it always goes wrong. If pediatricians always frequently annotate ages by month, and this get lost, like Melanie showed, from one step to the next, the number may stay, but maybe the unit is gone. If you use ISO 8601, then it is self-scoping for value and unit, right? So this is important and this is a continuous process where people develop standards, implement standards, test them, and so on. And but it's a very dynamic process, and it's very good to follow that. Some of this we try as part of GA4GH, where I'm a co-lead of the discovery work stream. Some of this we try to capture and support through efforts like the Schema Blocks initiative, where we look what do standards and products throughout GA4GH use as standards, and then tease them apart and document them and pull them and put them on these on the website and say, okay, here's documentation, here are the links to these original formats, and this looks then currently like that. So you have, for instance, a biosample definition, or you have a Curie definition, and so on. And where are they then? Where do they originally come from? Pointers to the resources where you can find them, and have this as a kind of exchange platform for looking at standards, and then also connecting them to who and where they are defined originally, to facilitate people to build their own products and using things other people in GA4GH used to, without just taking the whole bundle and saying I have to implement the whole phenotypics. Okay, so what have we learned as in our specific cancer genomics use case, implementing in our resources these standards we are partially involved in developing with? Beacon is very capable already to drive actually data-rich resources, and the filter paradigm has really proven itself as being super useful. There may be additions to that and future extensions like query logic and so on, a bit more complex. How do you do data aggregation and so on, which always some implementations will go forward beyond the standard, and then hopefully they will be fused in over time. Yes, use access to data, and who is allowed access to data has been mentioned before already. Duo, Passport, ELIXIR AAI important, and Schema Blocks is a good exchange place for looking things up. And if you see that the need for documenting other clearly defined standards there, please go ahead and contact us to push things there and keep this in dynamic environment. Now, if you do rich metadata annotations, then you can also do something like that. And this is actually, the figure is now in a publication we had this year. You can also look for like other analysts. We are looking now not at cancer genomes, but we are looking at where do the cancer genomes come from. And it actually was interesting if I looked at the H3 Africa project, which is a super cool project and highly organized with lots of different parts, but I didn't see much of cancer data focused there right now, and that's a pity, and we hope this will change over time, that researchers from everywhere in the world become interested and also doing supporting analysis in the cancer areas in Africa, because this world map basically shows where the samples come from, which have been analyzed in more than, so this is a view of more than 170,000 individual experiments. Where do they come from, and where have they been analyzed? And we did actually an analysis if this is correct. So we are pretty good on a country level. Not fine-grained resolution is not necessarily very good, but we can say, okay, some continents are great and their populations are greatly underrepresented. Now coming to the last several slides about how ELIXIR is now showing any other involvement of ELIXIR in this. Now, we talked about Beacon. I talked about Beacon and how we implement Beacon before, but the important point is not to only to extend Beacon protocol, but to make it something, the power through the network, the strength is in the network, and developed by ELIXIR Finland. We have now with the ELIXIR AI contribution and so on from Czech Republic and other areas, there is now ELIXIR Beacon, which already starts to have this high quality beacons which answer to the newest protocols and integrate them. So starting to build up a new more data-rich network of these beacons. And we hope that this will kind of slowly or the faster the better, lead to kind of, well, saying it, Internet of Genomics. This is a bit of a moniker, but one has to set the targets high sometime, because if you look at Tim Berners-Lee's first proposal about the World Wide Web development, there was a note by his supervisor that it is vague but exciting. And this was in 1989. Last slide. ELIXIR Cancer Data Focus Group is something coming up. This is something where our efforts of lots of other people involved in cancer area are also become part of and drive these cancer data exchange forward. And also now our focus is on human copy number variations. So ELIXIR has launched a human copy number variation community that I'm part of, and which also tries to see how these data types, not only from cancer, but now copy number variants from rare diseases and cancer. So we have also this connection to rare disease communities, how they all come together and are organized and driven forward by community of experts, which also have access to resources and also are very interested to make the topics of their research and their clinical interest and research interest available to wider communities and empower wider data use there. Some names here and some resources. Slides are up. There's my group. I have to thank my group, former and current members in my group at the University of Zurich and the Swiss Institute of Bioinformatics, with a special nod now to Pierre-Henri Toisson, who is a higher developer who's doing the new website front end, which comes out quite nicely. Beacon API leads Jordi, Tony, Juha, obviously Mark, my co-lead of the Discovery Workstream and Global Alliance, and then ELIXIR, especially Gary, but also David and Serena behind that, and developers like Sabella, who put lots of effort there, and the GA4GH, Melanie, Helen, and other people working with us together in the standards definitions. And last slide is always the Swiss slide. So thanks for having the time for this talk. Thank you.
Thanks, Mikael. That's a really great. There's a bit of feedback on your speaker. I'm looking at the open air platform and I don't see any questions. Are there any questions from the co-chairs or the co-panelists who'd like to ask Mikael something? So every time I see the complexity, Mikael, I'm really happy that you're one of our leads of the Cancer Data Focus Group. I think as we try and explore the ELIXIR position within cancer data, to have the expertise that you've just presented is really fantastic. If there are no questions, then I think maybe in the interest of time, we could move on. We have two technical presentations left, one from Babita and another from Shubham and Alex Knitz. I'll just highlight to you both, or all three of you for the both presentations, that we do schedule until top of the hour. So we've got 29 minutes left. If we need them, we can run over for 15 minutes, but let's hope that we don't have to use them so that people can stay with their scheduled agendas. But I highlight that you will not be cut off mid-sentence. There will be time to overrun if we really do need them. Let's hope we don't. But for now, Babita, if you're ready to present the use case on complex diseases, I ask you to share your screen.
Thank you, Gary. Yes, let me share my screen. Please remind me if I'm out, if I'm going out from, just a second. Please remind me if there's I'm moving, but I will try to move faster.
Okay, no problem. But of course, you're presenting the infectious diseases use case. We've had complex diseases, so I look forward to your presentation.
Thank you. Good evening, everyone. Thank you for the organizers to give me this opportunity to talk here. I would really like to make a disclaimer before that my background is not at all infectious diseases. What I would like here to highlight the implementation of Beacon that we did for the COVID-19 viral Beacon, virus data. And I would like to thank Michael, because you really wonderfully explained Beacon, so I probably will save some time there. What I'm going to explain here about Viral Beacon is how we developed. I will make a demo as well about the platform that we have developed in EGA in this group for finding variants, genomic variants on COVID on SARS-CoV-2 genome sequence. So if you see here, there is a genome sequence, and quickly, what are the variants? So they are the genomic alphabets, let's say, and the variants will be these. You see that if there is supposed to be A in that position, there will be a bit of mutation or a variant that is converting to T or G.
Babita,
So, yes.
we can't see your screen. Are you planning to share slides?
You are not seeing my screen?
No, I can't.
Oops, sorry, you should have. Sorry, now?
Yes, now we can see all of your window.
Oh,
and now we can see the presentation. Fantastic.
okay, thank you. So what I was saying here is the platform that we have developed for COVID, for SARS-CoV-2, to extract information from the viral genome. And what I was explaining here was this figure, which you see the genomic region of this virus and the variants that we are interested in. For example, if there is supposed to be A as in the main nucleotide, and it can be mutated to T or G. So this kind of mutation we can identify based on this Viral Beacon platform. I will make a demo quickly. Just wanted to say, but Michael already said about what is Beacon. The idea was to make a quick discovery of genomic data while keeping the privacy intact. So the previous Beacon, the old version 1, was that to tell you, user can query, for example, have you seen this nucleotide at this position? And the old Beacon was telling you yes or no. Then this is just how, if you could talk to Beacon, the difference now about Beacon 1 and Beacon 2. So version 1 would say you, yes, yes, there is this variant, but it will not tell you something more about it. In the Beacon version 2, what we are doing now is to make it more diverse, make it more responsive, to give you also information about, not just about the variant, but also about genotype, phenotype, metadata. So a lot of things has changed. I'm going to skip this. This is what has changed. But you can we can make different queries. We can ask different queries, not only about the genomic variant, but also about patients, for example, without jeopardizing the privacy of the patient. And we can ask about biosamples, metadata, etc. We can add filters to it. There are different schema versions as well as data access layers to extract these information, but I will quickly skip this. But yes, I would like to talk why Viral Beacon. We were in the beginning, we were very inhibited to jump into something that is not in our expertise, and none of us in the lab had any idea about virus genome. What was happening during those days in the beginning in March, I think we suddenly got shut down at the home, and then we started getting all of these pictures. For example, you see this one is from Hospital Del Mar, which is next to our lab. And it was our regular day-to-day, going up and there. And then we suddenly see this kind of pictures of doctors who are bringing patients outside, when they have started feeling better, to see them see. And so then also our institute started sending us mail to volunteer if we can for analyzing the patient samples, for example, or in another way. So then we thought, okay, can we then jump in? Can we do something with what we have? So we had Beacon then, and we were working on version 2. So then we thought, okay, it can be a good case for us for implementation and testing all of these filters and everything that metadata and everything that I was talking before. So with this idea, we started to take the risk. Those time also there were only consensus data available for the virus. So what consensus data is something that will show you most frequent variant at a genomic position. So then we had to develop also analysis pipeline for the raw read sequencing that was not available, because what we wanted was for with genomic variant, what we would like to do is to understand the variation at all, all the variants, not only the consensus or the most frequently represented variants, which in epidemiological studies it makes sense, but we wanted to in fact include all variants. So these are the different analysis pipeline for different set of data that we are using Illumina and Oxford Nano for raw read data, and we use consensus data from GCAD and from ENA consensus that were submitted. We developed the pipeline specific for these different data set, and then we added in the database to make it query. These are the samples that is when the data freeze of 4 August we had in the beacon from across 93 countries. And let me see if I can show you the demo. So this is how the page looks like now, which is you can search for the variant here, then some annotation about the information about the variant. You can get the frequencies represented for this variant in different data set and the metadata corresponding to it. So this will be, for example, you can search for the query. You can filter here based on countries, like host sex, host age, platform, geo-origin. You can search, for example, if you're interested only in America, Spanish data or something like that. There are other type of queries. For example, there is region query that can returns you specific interested regions. There are feature query that is based on annotation. So you are looking for certain genomic annotation. There is motifs. I have already opened it here. Yeah, so you can search for repetitive k-mers, for example, if you're interested, and it can tell you if there are mutations on certain k-mers. And there are different ways to find out more interesting variants or highly frequent or low frequency variants and such that. So please feel free to try. I will go back to the presentation. Okay, so I skip this. There are other ways, there are other examples how this can be used. For example, filtering based on a specific country or for certain genomic region that are interested, so that you are interested in. I will skip this. Why there's a use of this kind of beacon platform. Here is one use case to identify, for example, low frequency variants, which if you see what there is now, the data, most of the data that is available, for example, in GCAD, is a huge ton of data. But what it is happening there is they are showing mostly this kind of consensus data. So for example, you see this region, genomic region, will be shown as A, or that will be the most prevalent, most common information that we will find. And then what we lose is the small low frequency variations that individual, and then especially if we are interested, especially in those variant in different countries, for example, to associated with different metadata, etc., this information will be lost. So that will be one use case. Another, for example, will be to show, here I am, for example, I'm showing the differences in the number of variants between with consensus data and raw read data. So this slide is for intra-host variant. So intra-host variant will be, for example, here in the same, there could be mutations there could be different strains within the same person. And if you see the below plot, what consensus variant will show you will be these red variants, but low frequency variant intra-host variant will be these ones that the viral beacon can detect and tell you more information. Here is the plot for the allele frequency plot, sorry, to tell you that these low frequency variants are important. For example, you see most of these low frequency variants are non-synonymous mutations. So it is important to look into them and not to lose this information. Just to tell you that the number, for example, consensus data variants and the low frequency variant, we found almost 72% of the variants were novel and they were not in consensus data. Another example could be for these kind of low frequency variant to trace the evolution of these variants real time, as in when the data sample was collected to how it is responding and in different country data. And so on and so forth. There are many other uses. There are more information available on the page. Please feel free to take a look. And I hope that I convinced you that Beacon could be now useful for different other pathogens and diseases. And we have now we have the template to make it pathogen specific, or this was also a good opportunity for us to think about Beacon, not only human specific, what we were doing before. That was a mistake, I think. Now, after this use case, what we tried to do is to make Beacon more adaptable. We broadened the schemas to different categories. We added interactors, for example, host and pathogen information. So this has been very useful for us also to make it more inclusive, and I look forward to collaborate or to make it more useful if we receive some feedback to make it more useful for clinicians or researchers that are working in the field, and to give us feedback of how this can be used and utilized in a better way. With this, I would like to thank my team and collaborators who provided us data or who helped us with the data, the funders, the team that worked very hard in their confinement in their home with the laptop and some simple machine, but the work never stopped. So thank you everyone for listening, and I look forward to your questions.
Thanks, Babita. That's a fantastic presentation. I think it's really great to see the speed of response to the COVID-19 pandemic that's happened here. Before I look at OpenAir, I have a question about usage. So it's great that you provided a demo of the platform. Are you storing KPIs to see how many people are using this, or have you explored any collaborations based on this new use case that you've explored for Beacon? Where are the next steps?
Yeah, Gary, so we have added that, but however, we haven't made it. It is probably the first or second time that we are presenting it in public, the Beacon. First, we ourselves were not confident because every day there are new things to add. But now I think we are going to have a press release and to make it more, because now it seems to me that the interface and everything is at its place to be shared. So we soon look for, so the KPI is in place, but we will look, I mean, right now we don't have so much data.
Okay, well, there's around about 150 people online here, if I look at the numbers. So hopefully you'll get some people here that can test that and look at the data that's in that COVID Beacon and reach out to you with any questions that you would have.
Yes, that will be very helpful for us. Yes, thank you.
Are there any other questions from the co-chairs that they would like to ask to Babita? Okay. Babita, I would ask you to put the link to the COVID viral beacon into the platform. People can access it there, and as I say, they might follow up afterwards.
You mean I send the link? Okay, in the chat.
In that link, then people could access it and play around with it either now or later, and that might be a way to get some feedback on the platform. And then I think we'll move on to the final technical presentation, which is a joint presentation from Shubham and Alex. And I open the floor to you guys, to whoever's sharing the window.
All right, so I think you can see something on my window now.
Yes, we can see the presentation.
Yeah, okay, that's great. So welcome everyone. I'm Shubham Kapoor from CSC ELIXIR Finland, and today I'm going to present about ELIXIR Cloud and AI project, which is actually a joint project with multiple nodes and multiple ELIXIR teams involved. So moving on to the project, ELIXIR Cloud and AI project is basically an coordinated effort of ELIXIR nodes, which plan to deliver global alliance compatible cloud APIs to the users and to the end bioinformaticians who would be using platform for running some complicated workflows. The main motto of ELIXIR Cloud and AI platform is to enable federated computing environments which can deliver large-scale workflow analysis across international boundaries. And at the moment, ELIXIR Cloud and AI project as of whole leverages from different ELIXIR compute tasks, what we have within ELIXIR network. And ELIXIR Cloud and AI project, all in all, is also a Global Alliance driver project since 2019. As I said, ELIXIR Cloud project is a coordinated efforts with multiple national nodes involved within the project. So we have different ELIXIR nodes supporting this project. So mainly ELIXIR Switzerland, ELIXIR Czech, Germany, EMBL, EBI, ELIXIR Finland, ELIXIR Greece, ELIXIR Italy, and then we also get coordination support from ELIXIR Hub. And project is quite open, and we also encourage different ELIXIR nodes and other national institutes in case they are interested in joining the technical activities, what we have been doing in ELIXIR Cloud project. We have a brand new project website for our ELIXIR Cloud project. If you want to track the different activities or different roadshows and different programs and conferences in which we are involved. So we have a brand new project website telling about different activities in which we are getting involved lately. Other than that, we also have a GitHub organization. So it's basically an umbrella organization where we have almost all of our code repositories for the services what we are offering under ELIXIR Cloud and AI project. We also have some resources and test deployment page available on our website, which also gives you real-time statistics about different cloud APIs and their status. So which cloud API is currently working in which of the given ELIXIR node, and which cloud APIs are basically available for taking your workflows or taking different tasks for execution. So a founding stone of ELIXIR Cloud and AI project dates back to 2018. So it started with an ELIXIR implementation study, and that implementation study was all about to operationalize Global Alliance Cloud Work Stream standards. So Global Alliance is proposing quite many different cloud work stream standards, and our main focus was on key standards of Global Alliance cloud work stream standards in the form of WES, TEST, TRS, and DRS. So entire aim of implementation study, which started in 2018, was mainly to operationalize a workflow execution service, which will choreograph execution of different workflows, establish a task execution service network, which will actually start orchestrating different tasks or computational tasks among the nodes, a tool repository service where users or and bioinformaticians can save their tools and workflows, and then a DRS service which could act as a data repository for producers and consumers of data. So we started with this implementation study, and as I said before, we became a driver project of Global Alliance since 2019. And since then, we have been contributing to different cloud work stream standards. So we have been actively contributing in specifying cloud work stream standards for TRS, WES, TEST, and DRS. And then we are also adopting quite many different standards and different practices what Global Alliance is chalking down in form of different work streams within our ELIXIR Cloud platform. So For example, we are trying to implement ELIXIR AI, which again is implementing GA4GH passports and visas specification. The entire ELIXIR cloud platform, what we are trying to build is built across ELIXIR Compute Moonshot vision. So it's an high-level vision which aims in integrating user federation into local compute and data deployments, what we have within national nodes and within different ELIXIR nodes across Europe. And we plan to do that with the ELIXIR AI, which in turn is compatible to Global Alliance passport specification. Aim is also to provide some sort of national or regional workflow choreography engines in the form of workflow execution services. And then basically a network of task distribution network, which complies with Global Alliance test specifications so that actual tasks could be distributed among different ELIXIR nodes, and then also have an ELIXIR-wide data distribution network, which is again compliant to global standards like GA4GH DRS, and then have a curated repository for trusted bioinformatic tools and workflows in the form of GA4GH TRS compliant service. So as of now, we have multiple cloud services, what we have set up in the project and what we aim to use, what are being developed under ELIXIR. So under this project, ELIXIR Cloud and AI, we have successfully developed workflow execution service and task execution services, which are compliant to latest specification of Global Alliance West and TESS. These services are quite agile and are easily deployable on Kubernetes and are also deployable on individual servers using Docker Compose. Other than that, we are relying on different services from ELIXIR for our TRS and DRS endpoints. So for TRS endpoint, we rely on biocontainers. And for DRS, we are relying on RDSDS service, which is currently under development at EMBL-EBI.
For TRS, we are also going to work together with Workflow Hub, just to mention that.
Yes, yeah. So a Workflow Hub is also in question or actually one of the proper candidate what we aim to use for GA4GH test. So the state of current ecosystem, what we have for ELIXIR Cloud and AI project. So basically an researcher or bioinformatician can use our command line tools or our web portal to submit a workflow, which is written in CVL workflow language to our workflow execution services. Now our workflow execution services are regionally deployed in ELIXIR Finland, ELIXIR Czech, Italy, and Greece. I forgot to mention that a researcher needs to also authenticate himself. So that's the starting point. So only bona fide researchers who are able to authenticate themselves using ELIXIR AI are allowed to submit workflows. Now, when these workflows are being submitted to our workflow execution services, our workflow execution service basically submits computational tasks to appropriate test endpoint, which in turn are running on different national nodes and ELIXIR nodes within ELIXIR. So at the moment, we have different task execution endpoints running on Kubernetes clusters in different ELIXIR nodes like ELIXIR Finland, ELIXIR Czech, EMBL-EBI, and ELIXIR Greece, and so on. And then there are some upcoming nodes where we are also planning to have these task execution services deployed. Then when we were framing ELIXIR or when we were moving ahead with ELIXIR Cloud and AI platform, we also realized that there are some missing bits and pieces for within Global Alliance standards. And we figured out that in order to be able to actually run a federated analysis across multiple nodes, we need to also have some standards across task federation and workflow federation. So for that, we as ELIXIR Cloud and AI project, we are also trying to propose Global Alliance to have some sort of task federation logics also chalked down in these standards. And we are actually implementing some proof of concept services within our project, which we can showcase to Global Alliance, how such a distribution could take place on task level or at workflow level. So we are developing POC services in the form of protest and Distribute, which were basically Google Summer of Code projects as well for last year, students who were working with us, and then we are also continuing work this year for making these services more robust and actually to a stage where we can present it to Global Alliance and present our logics of what we could apply for task federations. Other than that, for long-term mission, we also plan to have some sort of workflow federation logics integrated in our platform as well. So not only task federation logic, and that is something on which we are working on at the moment. So we aim to envision a platform where we can easily federate workflows being submitted by bioinformaticians or end researchers. And depending upon their workflow types and compute backends, we could have basically full-fledged federation of workflows and task executions to multiple ELIXIR nodes or to multiple WES and test endpoints which are running in these ELIXIR nodes, which can actually run these tasks. Now, any platform is incomplete without having a real life use case or real life demonstrators. So we have successfully demonstrated some of the real life workflows or use cases. So we demonstrated rare diseases workflow on top of our ELIXIR Cloud and AI platform. So rare diseases workflow was basically a variant calling pipeline, and we demonstrated it using WES CLI, WES and TESC and Federated EGA pipeline. So entire pipeline was taking raw genomics data in the form of FASTQ files, and then finally producing unannotated GVCF files as the output, which were then getting encrypted using Crypt4GH, another utility what Global Alliance proposes, and then was finally submitted to Federated EGA environment running at CSC or ELIXIR Finland. Other than that, we also had multiple sequence alignment workflow. What we did in ELIXIR all hands meeting as an hands-on for the for the workshop participants, and this workflow was basically comparing two sets of peptide DNAs and RNA sequences, and was then generating an phylogenetic tree plot. We also aim to support real-life demonstrators coming within area of federated compute, and basically demonstrators which are compatible to Global Alliance Cloud Work Stream standards. So as a next step, we aim at supporting GWAS workflow on 1000 genome input, which is again basically a joint variant calling pipeline, which uses VCF and using Plink utility generates a scatter plot or Manhattan plot from the associated file. Now, this demonstrator is something on which we are working along with other GA4GH partners to get this demonstrator delivered by GA4GH plenary, which is at the end of this month. And we are basically working with other Global Alliance partners from Cloud Work Stream, mainly with Google and DNA Stack to have this joint demonstrator during GA4GH plenary 2020. So in case many of you are joining GA4GH plenary 2020, then you might see a real demonstrator working on ELIXIR Cloud and AI platform running then in collaboration with our other Global Alliance partners. Other than that, I think we were running short of time, so I tried to end up my presentation, and in case you have any queries, then I'm open to take any questions now.
Thanks, Shubham and Alex for the great presentation. Do we have any questions from OpenAir? None that I can see, so I open the floor to the co-chairs and co-panelists if they have any questions for Shubham and or Alex. So there's one here. How should data use cases, how should data use cases, okay, so how do, so there's one from Tommy here that I can't quite decipher just yet, but it says, how should data use cases, technical experts approach technical teams working on GA4GH Cloud and AI? Okay, so how do people reach out and get involved in this work and talk to you?
Yeah, exactly. What is the way to reach the technical expertise that is at the high level in this team? So what would be the best way?
So right now, so if you look on our GitHub organization, there's a big button. There's a badge to reach out on us on Slack, and there's a lot of information on like how you can contribute if that's your idea. And so this is more for development, and so for users, okay, there is a lot lacking yet because I think we are not there yet. So we have to work on this for the next year when we're really starting to roll out the stack to end users. And for people at nodes who want to implement services, so right now there's also the Slack space, but we are also working on a continuous delivery pipeline that individual nodes can subscribe to. So basically, each service runs their continuous integration on any change, which pushes a new container image, and then this would be alerted somewhere. It would be published somewhere, and like any node can subscribe to these changes and then update their deployment. Other than that, we have for each service, or almost all of them, we have like Helm charts for deployment available. So deployment should actually be relatively easy and relatively well documented. Yeah, but still, I have to say that we have to improve a little bit on the entry points, especially for end users. But for now, the best way to reach out to us is through the Slack space that there's an open invitation on our GitHub organization.
Yes, and other than that, we are also working closely with ELIXIR use cases community. So if there are some of the compelling use cases, what ELIXIR use cases community offer to us, then it acts as an internal driver project for us, so that we can make our platforms and APIs ready for those use cases and serve that particular community. So rare diseases workflow in past was such an example where we were being approached by rare disease community if we can build up a demonstrator what can execute their CVL workflow, and that's how the rare disease workflow got built up.
Right. Plus we have, I think the ELIXIR Compute mailing list would also reach us. We're reachable by email, and then there's these bi-weekly calls that we have that are open to anybody who's interested, and if you want to get involved. So those are other routes how you can get a hold of us.
Very good, thanks so much. There's one other question there, which I've not asked Shubham or Alex. Are your slides open access? Can they be shared? Because there's a question where someone would like to access these, and I've got a link to the Google folder, but are you happy that these slides are shared?
So, I mean, all these services and their source codes are open source, so they could be, of course, installed in individual nodes if you want to install them on your own local installation. If you want to use installations what we have within ELIXIR Cloud, then you should be part of, well, a node which basically bona fides use to use the service. So in practice, you should have an ELIXIR AI account, and then a node to which we have allowed access to. So that is the case.
Okay, fantastic. Thanks, Shubham and Alex, for the last presentation. So I appreciate that we're now 6 minutes over our allotted time. So I see no further questions in OpenAir. Are there any comments or questions about the session from the co-chairs or the co-panels lists? If not, then I have this final slide to present, which is to thank all my co-chairs, panelists, and of course the attendees. The numbers have stayed relatively stable throughout the 3 hours that we've been presenting. I think the open air platforms worked quite well for discussion. We've had some questions in there along with the Zoom questions. But if anything is unclear or you want contacts for anyone that's presented and you can't quite find it, please do reach out to me. My email address is there, so it's gary.saunders@elixireurope.org. And then one of the final things I want to say is at the bottom here, and that's a plug for our one of our next ELIXIR events, which is the Virtual Biohackathon Europe. So if you're interested in the work that's been presented here, or similar work that we have across many different use cases within ELIXIR, the Biohackathon Europe runs virtually this year from the 9th to the 13th of November. There are 40 projects where you can get involved and get your hands on some of the technical work that we presented here or other cases. And the registration is open until 18th of September. The link is there. It's free to register, and it'd be great to see as many of you there as possible. For today's session, I thank everyone once again for attending, and I'll bring the session to a close. Thanks all. Bye now.